Bacteria are small organisms that cannot be seen with the naked eye. They are found everywhere on Earth. For example, on your phone, on the sandwich you forgot about in your fridge 3 weeks ago, and even on your skin. The majority of the bacteria you encounter on a day-to-day basis don't pose a threat to you. Some are even helpful. Bacteria are used for the production of some of our favorite dairy foods like cheese and yogurt. Not only are bacteria present in the world around us, they can also be found in and on our bodies. For example, there are some bacteria that live in your gut that help you quite a lot. They can help with digestion, boosting your immune system, and altering your mood towards a more positive attitude, but some are bad. When a bacteria enters an area that usually it doesn't belong in, like from a cut or an open wound, it has an opportunity to do harm and make you sick. More specifically, it takes advantage of the space and resources given to it from its new environment. This involves the bacteria colonizing your body. In this process, the bacteria can release various toxins to harm and better invade your body. Once they have successfully invaded and colonized, bacteria can begin to replicate more clones of themselves. This is the ultimate goal of every bacteria on the planet, to successfully multiply and make as many clones as possible. Now the question is, how do we keep this harmful invader from using our bodies as potential breeding grounds? First off, your body has its own method of fighting back bacteria called your immune system. In general, your body's own defenses are enough to keep many potential invaders at bay. But sometimes, in severe cases like bad injuries or multiple infections, your body's immune system won't be enough to successfully fight off and kill harmful bacteria. Your body needs some extra assistance, and luckily, science has provided the tools to help us fight off infection. Whenever you get a bacterial infection, your doctor will often prescribe you something called antibiotics. Antibiotics are drugs that can help get rid of bad bacteria by interfering with a key component in the bacteria's life cycle. Although antibiotics are extremely helpful in getting rid of bacterial infections, too much antibiotics can be harmful and can lead to antibiotic resistance. Bacteria, just like humans, are all unique and have genetic material. Some bacteria have genetic material that codes for mechanisms that can protect themselves from the antibiotics presented against them. These resistance mechanisms can be shared and passed along to surrounding bacteria in a process called horizontal gene transfer. Horizontal gene transfer is defined as the movement of genetic material between organisms. There are 3 main types of horizontal gene transfer: transformation, conjugation, and transduction. Transformation is the process in which bacteria can pick up random floating DNA from its environment and reincorporate it into its own genetic material. Sometimes, the floating DNA can contain genetic material that codes for a resistance mechanism. The insertion of this newly acquired resistance gene can result in bacterial resistance against antibiotics. Thus, the process of transformation can result in the spread of antibiotic resistance. Conjugation is the process by which 2 neighboring bacteria exchange genetic information through direct contact. In this case, one cell is acting as a donor for genes and the other is acting as a recipient. The donor has to create a structure known as a pilus in order to successfully partition off genetic information to a recipient bacteria. This type of horizontal gene transfer differs from transformation in the fact that it relies on the physical contact between 2 bacteria for the exchange of genetic materials. Just like in transformation, antibiotic resistance genes have a possibility of being carried over to adjacent bacteria and generating antibiotic resistance in a given population. Transduction happens when a specialized virus called a bacteriophage infects a bacterial cell. When a phage reproduces inside of a bacterial cell, it has to make copies of its genetic material as instructions for making new viruses. Sometimes, instead of only including the instructions for making the virus can package the genetic material of its host into itself, leading to genetic material being indirectly exchanged between bacteria when new host cells are infected. In this process, just like in transformation and conjugation, antibiotic resistance genes have the ability to be exchanged between bacteria. With bacteria having many ways of sharing and spreading their resistance genes with surrounding neighbors, it is important to limit the usage of antibiotics. Too much bacterial exposure to antibiotics allows for transformation, conjugation, and transduction to speed up the process of antibiotic resistance. Continual exposure of bacteria to antibiotics can result in a high increase in multi-antibiotic resistant strains of bacteria. In order to combat the rapid rise in antibiotic resistance, we have a responsibility to use antibiotic prescriptions exactly as prescribed. This includes taking antibiotics only when prescribed and taking the allotted prescription completely, even if you start feeling better before you finish the prescription. Limiting the opportunities for bacteria to survive after being exposed to antibiotics limits their ability to develop new resistance mechanisms. By doing this, we can slow the spread and rate of antibiotic resistance in hopes that novel treatments can be made to outpace the evolution of antibiotic resistance in harmful bacteria all over the world.